Okay, here I'm going to describe one of our experimental setups that we have in Trinidad, set alongside a natural stream. And this is a site where we do replicate factorial experiments and can test ideas or evaluate mechanisms that underlie the sorts of things that we see going on in our natural streams. Part of the point is that our natural streams and the experiments that we do there, which involve introducing guppies, set up this grand interaction. You add guppies and then you ask what happens in response to the presence of guppies. But the data that you get are this very long and complex time series stream of information on many different variables. And it's hard to use that kind of information to make inferences about cause and effect relationships. And so what we've done is to set up what you see right here. This is a series of artificial streams. These are 8 different concrete channels, and each channel is subdivided into 2 different replicates. So we have a total of 16 replicate artificial streams in which we can do experiments. And 16 turns out to be a magic number for looking at cause and effect relationships. And I'll explain what I mean by that in a few minutes. If you look at me and look at the background, what you're looking at right here is an agricultural patch. It's something called Christophine. It's a very popular and profitable crop that's reared in Trinidad. And this is land that's owned by somebody who lives near the station, so we rent the space on the land from him, and the land is otherwise used primarily for agriculture that you see in the background. And then you can see some of the natural forest lying behind the agricultural fields. Now the way this works is we have this tank right here, and this tank is gravity fed from a spring that's somewhere up on the hill behind me. The spring normally would have outflow that would go into a stream. There's a natural stream flowing right here behind our experimental setup. And so before we came along, all the outflow from the spring went directly into the stream. What we do instead is we divert some of that outflow so that it fills this tank, and then from this tank we then feed water to all of our experimental setups. And each one of these channels is plumbed independently. It has a valve that we can adjust independently to adjust the amount of water that goes through the valve. And in that setting, we can do a number of different types of experiments. Now, one of the kinds of experiments that we do here is where we address whether or not there's a difference between guppies that are adapted to high versus low predation environments and the impact that they have on their ecosystem, on their surroundings. We know if we look at the shape of the guppies that there are important differences in the shape of the head and the position of the mouth, which in other fish suggests that they're different ecologically, that they feed differently in whether they are pecking along the bottom or feeding out in the water column or picking prey off of the surface, but also in other fish it's suggested that they have different diets. And what that would mean is that as a guppy, a high predation guppy adapts to a low predation environment, it might actually change the way it utilizes its resources, and that could in turn change the structure of the habitat in which they're found. And this is part of what we're looking for in the study, because we're interested in the interaction between ecological and evolutionary processes. So the idea is that as a guppy evolves and adapts to its new habitat, it changes how it uses the habitat, which could change the structure of the ecosystem, which then in turn could feed back on the kind of selection that the fish experience. But in any case, in this kind of setup where we have 16 artificial channels, we can do experiments in which we contrast guppies from high versus low predation environments, and in which we keep them at high versus low population densities, because we know that guppies from high predation environments live at lower population densities than those from low predation environments. And then we could, for example, manipulate light level. We could have high and low light level treatments by placing a shade canopy over the stream. And we know, for example, that the high predation localities have higher light levels and higher primary productivity than do the low predation localities. So we could do a 3-way experiment: origin, site of origin of the guppies, population density, and light level. And that results in 8 different treatments. It's 2 by 2 by 2, which is 8. And we have 16 channels so that we can have replicates of each of those 8 different treatments distributed at random along the channels. And then we can do a study that's in partnership with our ecosystems ecologists. And they would evaluate what the ecosystems are like at the beginning. And we could demonstrate that at the start of the experiment, we had true replicate streams, that the structure of the ecology within each of these channels is a reasonable replicate of what you would see in a natural ecosystem. And then we could apply our treatments which would be to put in guppies at different population densities from different sites of origin, and then we could continue to monitor the environment and look at how things changed over time and ask whether or not guppies from high predation environments do indeed have a different impact on the ecosystem than those from low predation environments. We've already finished some of these experiments. We found that in fact they do, that the high predation guppies do what you would infer they would do from the differences in the head shape. High predation guppies prey more on invertebrates, on insect larvae, and we can show that by what we find in their stomachs. We also can show it by the abundance of invertebrates in replicate channels that either do or don't have high predation guppies versus low predation guppies. And then we can do other things to evaluate the impact of these two different types of guppies on the structure of their ecosystem. And so we've already been able to pose hypotheses and test them using this kind of experimental setting. This setup that you're looking at right now is an experiment that's being done on the killifish, Rivulus hardi. And in this case, what we're modeling is not an active stream, but instead we're modeling a backwater, the pools that form on the sides of streams when they overflow, because that's a favorite habitat of the Rivulus. And so what you're seeing now is not a clear running stream with a gravelly bottom, as much as a still pool which would have a lot of sediment accumulated in it, which is the kind of habitat in which Rivulus find themselves. But also one of the things we're looking at in this particular experiment is the interaction between Rivulus and guppies. So some of these containers will have just the killifish, some will have the killifish plus the guppies, and in that kind of setting we'll be able to ask some questions about what the impact is of guppies on the killifish, since we have other data done on natural streams in which we have an implication that there are such interactions. But the main point is to understand how we can get at mechanistic interactions in this particular kind of setting. We already knew that high and low predation guppies differed in body shape. We knew that if we went out to a natural locality and collected high predation guppies and looked in their stomachs, that what you found in the stomachs was different from what you would find in the stomach of a guppy from a low predation locality that was collected and preserved on site. But we don't know if that's because the guppies differed in how they foraged, or if there were differences in what kind of food was available for them to eat. Here we can put them in channels where we know that what's available to them is identical, and then we can ask more critically whether or not there really are differences between them in their preferences, and find out whether or not those differences in preferences feed back on what the structure of the ecosystem is. And that's a step that we've already accomplished here. We've shown that the guppies really do differ in what they prefer to eat, and that a consequence of those differences is that you get a change in the structure of the ecosystem. High predation guppies eat more invertebrates. Invertebrates happen to feed on algae that grows on environmental surfaces, so that when a high predation guppy is there, you actually get more algal accumulation than when a low predation guppy is there, because they're eating the things that eat the algae, and by removing the invertebrates that eat algae, there's an increase in the algal abundance on the environmental surfaces. So that's the kind of mechanistic question that we can ask here, and in that way we can complement the kind of information that we get when we look at the focal streams and we collect a time series of data over a prolonged period of time, over a period of years. And as I said, we learn a lot about what happens, but we don't necessarily see the mechanisms for what the underlying interactions are.